what are some of your highlights scientifically from your own research? Because you were very, very active yourself. You were a leader, not just in leadership roles, but also in research.
Yeah, I think I can remember a few times being particularly excited about getting to a point in a project which was an end as defined by this can now be published, as it were. And one of the first ones was, if you remember, when I first went to EMBL, I worked on the transcription of US and RNA genes, and they had promoters, and they have it's a very interesting problem. They have promoters in each eukaryotic species. They all have the same set, more or less, of USNRNAs that are involved in splicing, in the removal of introns. They have SNO RNAs, which are involved in ribosomal RNA processing. And they have a few other small RNAs that are involved in things like making the 3' end of histone messenger RNAs, etc. So they all have this array of something like 27 or so USNRNAs. And all of these USNRNAs are short. They're very highly conserved between species in terms of the RNA sequence. And they have promoters which are different from all the other promoters in the cells that they're produced in. And these promoters, however, they evolve in two ways. One is that some of those RNAs are produced by RNA polymerase II and some by RNA polymerase III, but their promoters are extremely similar. And there are different ways of switching between RNA polymerase II and RNA polymerase III. In plants, for example, you do it by changing the distance between identical promoter elements. You just move them further apart, and you go from pol 3 to pol 2. In the case of the vertebrate USNRNAs, which is what I worked on, U6 was the one that was transcribed by RNA polymerase III, and the promoter was identical to those for the other USNRNAs that were involved in RNA processing, except that it had a Tata box centered at -25, which is the definition of messenger RNA polymerase II promoters. But U6 was a pol 2 promoter. U6 was a pol 3 promoter, sorry. Messenger RNAs are pol 2. And I remember the day when we discovered and proved that it was the Tata box that made the U6 promoter a pol 3 promoter rather than a pol 2 promoter. It was such an unexpected result. And I remember running along the corridor and yelling to probably you, to Riccardo Cortese, to Roberto Di Lauro, to Henk Stunenberg, to everybody who was around. David Tollerby was around. This is amazing. Look at this. It can't be true. And it was that was very exciting.